Adelme Bazin. Adelme is from the lab GEM. He will present the result about the pangenome analysis to discover genomic islands. This is the highlights of the paper that was published in Bioinformatics last year. And Adelme, the floor is yours.
Thank you for the introduction. Do you see my presentation?
Yes, perfect.
Okay, awesome. So hello, I'm Adelme Bazin. I'll be talking about panRGP today. So it's a pangenome-based method to predict genomic islands and explore their diversity. Nowadays, when you conduct prokaryotic genomic studies, you tend to rely on the comparison of hundreds to thousands of genomes of the same species when you're studying a species. Those genomes display a very high diversity in terms of gene content. Most of this diversity comes from horizontal gene transfer. And between 5 to 40% of genes between two different genomes of the same species are different. So this very high number of data and of differences gave rise to the concept of pangenome. A pangenome is a union of sequence entities shared by genomes of interest, usually from a given species, but it can be also from a genus or a phylum. It also gave rise to new data models, moving away from a reference genome that only has specificities of its own, and towards reference pangenomes, which encompasses the overall genomic diversity of the group that you're studying. So you can build the pangenome at different levels. So you can build a pangenome using sequences only if you are interested in a variation between different genomes, for example. Or you can also build a pangenome by looking at the gene content, which is what we do. So our goal is to study the diversity of genes shared by a group of individuals, usually a species. In this example, you have 4 genomes. The rectangles represent genes. Genes have the same color in different genomes if they are homologous. When you conduct a pangenomic study, you can partition the pangenome, which means that you can classify the different gene families into core or accessory genome. A core genome will include all genes that are present in all individuals of the group that you're studying. So here's red genes, for example, are present in all genomes. So they are core. The accessory genome will include genes that are present in not all genomes. So it can include rare genes or genes at intermediate frequencies. So this was the partitioning scheme that was used originally in the first pangenomic studies. However, as you can notice, it's not very stringent, as if you have gaps in your assemblies, for example, you might be missing genes in your assemblies, but the genes are actually present in the genome that you're studying. So it will drop the core genome artificially. So for this reason and some others, we use another partitioning scheme that was introduced in the Kunin and Wolf 2008 paper. The partitioning scheme goes as follows. We have the persistent genome, which is sort of a relaxed core, which will include genes that are present in all or most genomes of the pangenome. So it will include the gray gene, which was not part of the core genome earlier. The shell genome will include genes at intermediate frequencies. So present in this example, it will be present in 2 genomes, for example. And the cloud genome, which will include rare genes. So to realize this partitioning, we developed a tool called Pangolin. This tool relies on a pangenome graph. So how we build this pangenome graph, we use annotated genomes. We cluster all the genes together to build gene families. Then we build a graph where nodes are gene families and edges between the nodes are adjacencies along chromosomes. So here, the blue and green nodes are close together in the graph because they are linked in the genome. Then we partition the graph into the 3 partitions. So most other approaches rely on hard-coded thresholds that are usually picked because most people use the same thresholds. Here, we chose to use a statistical partitioning method called the neighborhood expectation maximization. It relies on a binary mixture model combined with a Markov random field to produce a partition pangenome graph. And so here is a representation of an actual pangenome graph. So this one was built from 3,000 genomes of Acinetobacter baumannii. What I can illustrate with this representation is that gene repertoire changes are concentrated in some hotspot locations and usually driven by horizontal gene transfer. In this zoom that we have here, you can see that there are two paths in orange for the persistent genes. And from those two paths are multiple cloud or shell paths that branch out. So those changes concentrate in genomic islands. And genomic islands are specific areas of genomes, of microbial genomes. They are often associated to tRNA genes to integrate to insertion sequences. And they have been of very high interest to microbiologists for the past 20 years because they aggregate many strain-specific adaptations, such as environmental adaptations, pathogenicity islands, antibiotic resistance mechanisms, and from time to time, secondary metabolites as well. Those genomic islands display a very high turnover in terms of gene content, as I illustrated on the previous figure. And they also have a composition bias, meaning that they have a different GC content compared to the rest of the genome, sometimes, but not always. Because of the reason, we expect genomic islands to consist mostly in shell or cloud genes. So we chose to design a method based on the pangenome partitions to try to find those genomic islands. The method goes as follows. So we use the pangolin pipeline that I introduced earlier. Then we project the partitions on the genes. So we annotate each gene based on the partition of its gene families. Then we compute a score along each of the genomes that we are interested in and we want to predict genomic islands in. The idea behind the score, which I will not detail here, is that stretches of shell and cloud genes will have a high score, and stretches of persistent genes will penalize this score. So in the end, what we expect to be genomic islands will be the regions with the highest score in the genomes. We benchmarked this approach against published datasets. Datasets that were published originally in this 2008 paper and updated recently in this 2018 paper. The first dataset is a comparative dataset. It was generated from Comparative Genomics, and it includes 81 genomes from 36 different species. What we did with panRGP is that we built 36 pangenomes using all available genomes in RefSec for all the species of interest. Then we predicted the genomic islands with panRGP and then computed the benchmark. We compared the scores of panRGP with many tools that are available in the literature to predict genomic islands, and we noticed that panRGP came on top with most metrics. So we were very happy about that. The second dataset is the literature dataset. It includes genomes with published descriptions and published bioanalysis of genomic islands. It's much smaller. It includes 6 genomes among 6 different species. So we built 6 pangenomes using all available RefSec genomes again from those 6 species. And once again, panRGP came on top with most metrics. So thanks to this benchmark, we can say that panRGP detects genomic islands very well. However, RGPs, regions of genomic plasticity, are not just genomic islands. They can correspond to both gains or losses of genomic regions. So if some regions are lost differentially in your genomes of interest, those regions that are kept in a few genomes of interest will be predicted as RGPs. They will also include plasmid sequences. So this is because plasmids are rarely kept in all the genomes of a species. So you will predict them as RGPs. And depending on the definition that you use of a genomic island, some may be persistent within a species and thus not identified as a region of genomic plasticity. I will illustrate that now with this Salmonella Antholica genome. So in this representation, you have the entire genome, which is drawn. The different lines correspond to predictions of some of the tools that are used to detect genomic islands. In red, you have panRGP. In black, you have the literature reference genomic islands. And I will zoom on one region in particular. So this is a pathogenicity island. It was described as such in the papers that described this genome. And it's of interest because it carries multiple virulence determinants for adhesion and entry into cells. And as you may notice, absolutely no tool, not panRGP and not any of the other tools, predicts this area as a genomic island. This is because it's defined as a genomic island because of the functions that it carries. However, it does not display any GC content variation, and it does not have any integrase genes. And all genomes of Salmonella Antholica that we use to build the pangenome, most of them at least, include those genes. So for this reason, it's not predicted by any other tool. So now that we saw that we were capable of predicting genomic islands, we might be interested in studying genomic islands that are found in the same persistent context. So those regions are called spots of insertion. As I said earlier, most gene repertoire changes are gathered into few hotspots. And in this example, which is a very well studied hotspot in E. coli, you see rectangles, which are groups of genes. And you see that in different genomes, you can have many different groups of genes, even though it's the exact same place in all the different genomes. So we define spots of insertion as persistent, as genomic islands that are flanked by the same genes, even if they don't have the same gene content. So since we could have so many genomes as we're building pangenomes, and since we could detect genomic islands so well, we decided to try to detect spots of insertion using pangenomes. So the method goes as follows. What we do is we take all the genomic islands that we predicted with panRGP. Then we look at the flanking genes that are persistent to the genomic islands. We compare the flanking genes together as ordered sets. If the ordered sets are similar or if they're partially overlap, we consider that there is a match. And we will build a graph where each node is an RGP, and we'll add an edge in the graph if both of the borders match. We do that for all RGPs that we predicted in the pangenome. If only one of the border and not the other match, we will not add edges. In the end, the spots of insertion that are predicted will be connected components in the graph. So we do not have benchmarks to test our method on a large scale. So what we did is that we looked for the LEX hotspots that I illustrated earlier in a metagenome assembled dataset. So we built a pangenome using 1,400 genomes from the Bazin et al. Dataset. Then we looked for the hotspots by looking for the persistent genes, which we found. This representation, so in this representation, each node is a gene family, and you can see colors depending on the partition where the gene family is assigned. This hotspot is found in about 400 of the 1,400 genomes. So we are very happy to be able to find it and to see that our method was capable of reconstructing the spots. So as a take-home message and in summary, I presented today methods of comparative genomics to detect genomic islands. We use pangenome partitions as a proxy for comparison. This way, the computational load increases linearly with the number of genomes, so we can work on tens of thousands of genomes. We can study genomic islands found in the same persistent context by clustering them into spots of insertion. That way we can study how the region evolves throughout a species. And all of those methods come along with a softer suite, which makes subsequent analysis easier. We can output many different file formats, which can be used with other softwares. This is available on GitHub. It's installable through Bioconda in the Pangolin software suite. It's also available in the microscope platform if you have genomes in this platform. Thank you to all the people that participated in this project. And thank you for listening. If you have any questions, I'll happily take them.
Thank you very much, Adam. It was very interesting. So yeah, same. Waiting for a question. I will shoot first. You are presenting a method that is working very well to me, and but it's based on the score, and you didn't have the time to explain a bit the score. And I was wondering, so the score is obviously the score is okay because it's working. But
Yes.
I was wondering if you guys check the sensitivity of the score and how much you gain or you lost if you move it a bit, and maybe how you could I don't know if you have other thoughts, especially to discover maybe missing genomic islands that were passing through the net. And so I was wondering if it was the point where you could maybe improve the method, or if you say no, it's okay, we cannot do better, and this is another game that we have to play.
So it's most likely possible to do better. We did not. I mean, we tested a few parameters, but we did not extensively try to adapt the parameters to the benchmark, because that would be cheating pretty much. But it's probably possible to do better. The scoring as it is done worked very well, so we chose to keep it as it is. But depending on the diversity of your data set, it is very possible that having different parameters can improve or decrease the sensitivity of the method. Yes. It's very possible. It's also possible that having different approaches can be better. I don't know.
You know, my point was not to change what you did already. Okay,
Yeah.
just for the perspective and to move on. Yeah.
Yeah.
There is a question from Leonard in the chat. It's a question about the pangenome graph. Are they always made of unique connected components, or can they be split into multiple subgraphs if the reference genomes are used are fragmented in multiple contexts?
Okay, so that's yeah. So so the pangenome graphs can be made of multiple components, but usually there is a single big component which corresponds to the genome. And sometimes you can have small components which correspond to plasmids. So I don't have a good example in this figure, but in other pangenomes we saw this. We saw this in other pangenomes. For the other question, usually, so if we use fragmented dataset like MAGs, we still find a unique connected components usually with maybe a bit more smaller components which can correspond to plasmids or to contamination, for example. But usually we have a single big component which is the main genome.
Okay. And there is we take the last question from Veronique. Did you characterize genes in different classes, persistent shell low, and for example, housekeeping genes? Where did you find them?
Okay.
And,
so indeed, we tried to look at functions that we could find in the different partitions, and indeed, we expect housekeeping genes to be found in the persistent genome. There were some classes that were enriched in the shell or cloud partitions. I remember now that genes that are typically related to antibiotic resistance are found mostly in the cloud genome, or sometimes in the shell genome, but very rarely in the persistent genome, for example.
Okay, thank you. Thank you very much for all the three speakers. That was a very interesting session. At least to me, it was very interesting. It's time to coffee break, and see you there in the Zoom later on. Thank you again.
Thank you.